Hello, I'm Howard Wetzman. I'm the primary addiction doctor here at Townsend. This is the Biology of Treatment lecture. In the last lecture on the biology of addiction, we discussed the nature of the illness, where it is in the brain, and why the drug you use really doesn't matter. In this lecture, we'll talk about how the biology you learned tells us how to treat the illness and relieve your symptoms. We'll talk about the order of treatment and how we'll measure your progress. Finally, we'll talk about how the changes made in treatment prepare you for a recovering lifestyle so that you can maintain the benefits you gain here. But first, let's spend a few minutes going over what we learned last time. In the last lecture, we saw that addiction is a chronic, biologically based illness that has symptoms which are independent of drug use. We saw that just like all other chronic illnesses, addiction gets worse with age and doesn't have a cure. But in spite of that, addiction has a very good treatment that can lead to resolution of the symptoms. No one has to live with untreated addiction anymore, and no one has to die of this disease. We just need to review one more thing before we get on to the biology of treatment. We're going to go back to the basic cartoon of the compulsive use circuit of the brain. Remember that when the enkephalin receptors on the ventral tegmental area get enough enkephalin, the ventral tegmental area releases normal amounts of dopamine. When the dopamine receptors have enough dopamine, we have mental focus, motivation, enjoyment of enjoyable things, and the ability to make attachments with others. Also, when there's enough dopamine tone at the nucleus accumbens, it sends a serotonin signal to the frontal cortex, giving us a feeling of well-being. Now, there are many different ways and many different places in this cartoon where we'll treat symptoms of addiction, so we'll show you each one. First, we'll start with the dopamine level at the dopamine receptor. Let's get rid of the rest of this for a while and concentrate on the area around the dopamine receptor. Over on the left is the nucleus accumbens. Those 4 purple blocks you see are the dopamine receptors. Each one of those hexagons in that circle down below is a molecule of dopamine. That circle represents a sort of bag around the molecules called a vesicle. Toward the top you see that kind of tunnel in the wall of the ventral tegmental area. That's called the dopamine transporter. The dopamine leaves the cell, gets to the dopamine receptor, and is sucked back into the ventral tegmental area to be recycled through the dopamine transporter. The dopamine is released by the vesicle moving to the left and merging with the cell wall, making a hole for the dopamine. Once the molecules of dopamine are released into the area between the cells, they're free to go to the receptor, get sucked in by the transporter. Notice that of the 7 molecules of dopamine we've drawn here, 5 have already been sucked back into the ventral tegmental area and repackaged in another vesicle. 2 are still bouncing around, touching receptors, bouncing on and off. They represent the baseline dopamine tone. Well, this is the basic dopamine tone, but what if you had fewer dopamine receptors? The dopamine tone would be lower, because the dopamine tone is set not just by the number of dopamine molecules moving around between these cells, but how many times and how frequently they're hitting how many dopamine receptors. So the fewer dopamine receptors, the lower the tone. The less number of dopamine molecules, the lower the tone. The less time those dopamine molecules have to hit the receptors, the lower the tone. But in this case, when someone doesn't have as many dopamine receptors as normal, but is making normal levels of dopamine, we can give them a medicine that blocks that tunnel, that reuptake tunnel. So now, some of the molecules of dopamine, instead of getting sucked up by the ventral tegmental area, are bouncing off the medicine and back into the area between the cells, where they have another chance at hitting some more receptors before getting sucked up in general. So they're sort of like the vacuum cleaner running with less power or slower. So now we end up not with 2 out in the cell doing their work and 5 getting sucked up, but 5 outside giving us dopamine tone and only 2 getting sucked up. So let's go back to the original cartoon of the reward system. Normally, when the nucleus accumbens is getting enough dopamine signal, it's sending a good serotonin signal to the frontal cortex. But what if there's something wrong in the nucleus accumbens so that even if it gets enough dopamine tone, it doesn't send a good serotonin signal to the cortex? Then we could block the reuptake of serotonin just like we did with dopamine in the previous slide, which would raise the amount of serotonin. That would really work well. The only problem is that the longer the serotonin is increased, the lower the dopamine release. So to increase the serotonin makes people feel good for a little while, and then after a while feel worse. So we've looked at how we treat a problem with dopamine and a problem with serotonin. Now let's concentrate on the part of the reward system that releases enkephalin. We'll zoom in right to the point where the nucleus accumbens sends enkephalin to the ventral tegmental area. Let's zoom in even more, close enough to see a single enkephalin receptor. And you can see how well your own brain's opioids, enkephalins, fit in this enkephalin receptor. But some people can have an abnormal enkephalin receptor where the enkephalin doesn't fit so well. But if such a person took an outside opiate like OxyContin or heroin, it would fit much better. Such a person wouldn't normally feel well unless they were on one of those outside opiates. But those outside opiates aren't very good medicine. What we want to find is a medicine that will turn the receptor on just enough to feel normal and not bounce on and off. It would also be wonderful if the medicine could protect people from outside opiates if they took any. And there is such a medicine called buprenorphine. Notice the buprenorphine can fully engage the receptor at 2 of those notches because it can turn on the receptor part of the way, just enough to feel normal. And notice how it grabs onto the whole receptor, not just fitting in lightly, and it sticks there, which is exactly what we want. Because if the receptor's covered with buprenorphine, this is what happens if someone takes an outside opiate. The opiate molecules will keep bouncing off the buprenorphine trying to get to the receptor until they kind of get tired and go away. There's always one that doesn't get the message. Now we go back to our original cartoon. We talked about how we treat problems with dopamine, serotonin, and enkephalin. But remember, there's more to this cartoon than we have drawn here. Let's move this down again and make room for the amygdala. When glutamate from the amygdala hits the ventral tegmental area, it sets off that positive feedback loop, causing a dopamine high, followed by a dopamine crash, which can cause craving and drug use. So the idea here is to block the glutamate receptor on the ventral tegmental area. Now the other treatments we talked about are different. They were all aimed at increasing dopamine tone or increasing the signal from the nucleus accumbens. But even a brain with normal dopamine tone can get triggered by a cue. And so we use glutamate blockers to decrease the likelihood of a cue starting a craving or drug-seeking behavior. So let's say we've restored normal dopamine tone and normal signal from the nucleus accumbens. The person either doesn't get any cues or we've covered their cues with a glutamate blocker. But sometimes, for some people, this feedback loop is too easy to trigger off. It doesn't just take a hit of drug to trigger it off, but sometimes even a lesser stimulus will trigger it off. And that means that even a lesser stimulus would cause a dopamine high and a dopamine crash. And the risk there is that the dopamine crash, even from a normal stimulus, might create craving for the next drug. So there's one more place we can intervene if we need to. And that place is at the GABA receptors on both the nucleus accumbens and the ventral tegmental area. GABA is just another neurotransmitter, but this neurotransmitter is used to slow things down in the brain. So if we increase GABA tone, what happens is the dopamine high isn't as high, and the dopamine crash isn't as low, and it's less likely to set off craving for the next drug. There are a couple of different types of medication we can use to increase GABA. The first kind are the anti-seizure medications. The second kind is a single muscle relaxant called baclofen. Both the anti-seizure medicines and baclofen increase GABA, and they can do it safely even in somebody with addiction. So let's summarize. If somebody isn't making enough dopamine because their brain's enkephalin receptors aren't receiving their own enkephalin signal, we can give them a normal kind of signal with the medicine buprenorphine. If somebody makes enough dopamine, but there's a problem with their dopamine being seen at the nucleus accumbens, we can raise that tone with another medicine that blocks the reuptake of dopamine. If the dopamine tone's okay, but there's something wrong with the nucleus accumbens and it can't send a good serotonin signal, we can strengthen that signal by giving a medication that blocks the reuptake of serotonin. If all those bases are covered, but the person's still having cue-induced craving, we can try to block the glutamate signal at the ventral tegmental area. And finally, if the feedback loop is getting triggered too easily, we can increase the GABA tone with a couple of different kinds of medicine. This order isn't necessarily the order everyone gets treated in. Everyone's treatment is designed especially for them depending on their symptoms and what the barriers to their treatment and recovery seem to be. I hope some of your questions have been answered about treatment, why we use medicines the way we do, and what we should expect from using medicines. If you have any other questions, please contact your treatment team.